Okay, good morning, everybody. So before I get started with any anatomy and physiology, I have a few announcements. One is that there is a biology colloquium this week, Thursday at noon in CST 103. And two students will be presenting their summer research projects. Aidan Johnson-Shukair will present his research at the University of California, San Diego, and Kai Win will be presenting his research in Germany at the Max Planck Institute in Frankfurt. So protein biochemistry is going to be the theme of the day. Two different protein systems. Anyway, pizza as always, and you're invited to the colloquium. Other announcements. Let's see. I'll just go backwards here. Okay, so there are a number of epic things happening. So one is Karen Mager is going to be leading an epic research project to Alaska, and she's having, mid-July to early August, and she is having, she must be having an information session, but it's not on the slide. So you'll have to ask Karen about the information session related to her epic. John Iverson is doing an epic May term, I believe, and he will be having an information session October 4th, noon, CST 103. I think that's next Thursday. And let's see, finally, Brent and Wendy will be leading an epic program on animal behavior and conservation in Tanzania, in East Africa. And again, there will be an information session about it on October 4th. So I believe there are 3 different epics going to be presented on Thursday, October 4th at the noon pizza slot. Okay, so if you haven't already used your epic, here are some biology options available to you. Okay, so now we'll turn to anatomy and physiology. So what we're doing today is transitioning from the cardiovascular system to the respiratory system. So a whole new system. So there's a handout here of the key concepts for this section, and as always, a handout of today's selected slides from today's PowerPoint deck, and more clickers if you've lost yours. No excuse for not having a clicker in your hand today. Let's talk about the exam for a moment. The exams are graded, but I can't hand them back yet, but I can hand them back this afternoon. So if you want your exam this afternoon, come find me in the Cell Phys lab after 1 o'clock. The Cell Phys students will be working on their poster project, it turns out, and I'll be available to hand back your AMP exam there if you're interested. If you just want to know your grade, it's on Moodle. Let's see. Lauren's not here, right? Okay, so I can talk a little bit about the exam. I want to say one thing, that the last question, which was the pressure volume loop, remember the one that had pressure on the x-axis and volume on the y-axis, and you were supposed to label the loop. That was a challenge question, and I was very impressed with how well most of you did with it. Honestly. So, and to accentuate that, on Saturday I was giving a heart AMP class to alums for homecoming, and there was a cardiologist in the room, and there was someone who had gotten an MD PhD from Hopkins, which is, he was not an Earlham alum. He was in a relationship with an Earlham alumna, but he was a MD, PhD. And when he heard that I challenged you to interpret the PV loop without ever having seen one before, he was really impressed. He said, you know, because he went to medical school, he knows what these things are, and he knows how challenging they can be. And he was really impressed that I would throw this at you in an exam setting. So, and he was also really impressed that, you know, that I said that you did very well with it. So, congratulations to you. Okay, respiratory system. So, you know, we have a respiratory system for many purposes. The three main purposes are here. Oxygen, providing oxygen, removing carbon dioxide, and one that you may not be thinking about, but it is just as important as oxygen and CO2, is pH balance. So the respiratory system works with the kidneys to control pH. And so this will be a link that we'll have to build as we get into the renal system later on. So, okay, I think I've asked you this question before. Why do we need oxygen? Okay, for cellular respiration. Why do cells need oxygen? So we're going to be talking about anatomy and physiology of respiration, but of course underneath that is cellular respiration. Why do cells need oxygen? There it is, the final electron acceptor in the mitochondrial electron transport chain that was needed for building up the proton gradient that was needed for the ATP synthase to make ATP via oxidative phosphorylation. And since we're aerobes, and we're not anaerobic, we need aerobic respiration. We need oxygen in order to perform the necessary level of ATP synthesis. Okay, so that one's pretty easy. Why do we need, what is the source of carbon dioxide? Let's go back to self-is. Where in your metabolic pathways do you generate CO2 that needs to be gotten rid of? Krebs cycle. And pyruvate oxidation, right? So if you said glycolysis, the answer is no. Glycolysis does not generate CO2. Glycolysis generates pyruvate. Pyruvate then is oxidized to acetyl-CoA, and that process generates CO2. And then acetyl-CoA enters and runs around the Krebs cycle, and that process of oxidation of acetyl-CoA generates CO2. Histology. Does somebody want to define what histology is compared to anatomy? What's the definition of histology? Because that's one of the things we'll be talking about today. It's microscopic anatomy. It's using microscopes to identify different cell types in an anatomical system. We don't do a lot of histology in this class, but it's there. Okay, so here's a slide from your textbook with some, but not all, of the terms that you will need to learn. Your anatomy coloring book is helpful here. The key concepts list gives you a list of anatomical terms to know. The quiz, which will be available on Thursday and is due on Monday, not Sunday, but due on Monday at 11:30 p.m. One extra day, is mostly anatomy. And it will cover the material from the coloring book and the material that we cover in classes today and Thursday. So it will be available at the CST desk on Thursday, and it's due Monday night, 11:30. Okay, so we're focusing on anatomy. Here are some key concepts. I'm not going to go through it because I don't like teaching anatomy. Well, I do like teaching anatomy, but mostly in the lab, not here. Here's a game. Some of you have already played this game. Why is it doing that? play this game. Okay, so here is a laser pointer. You're allowed to pass. And the idea is you tell me which of these buttons is which of these names. And then I will make it happen. Hopefully we'll hear a happy ding sound to tell us that we got it right. That one is the trachea. There's the sound. Okay, go ahead, everyone. Okay. Well. So this is a form of learning. It's not a form of testing yet, but
they all should be in the picture.
They're all in the picture. Each dot has a name, and each name has a dot.
So the top one is the larynx.
Okay? The larynx. Who's that?
I want to figure out what apex is. It's either this one or around at the end. Apex.
You have to choose one.
I'll choose top one.
Okay? Apex, the top, is right. Apex, you know, the pinnacle, the top. The apex of the heart is actually the bottom, but whatever. Inferior lobe. That's a pretty, that sounds like you're not sure, but that's a very good guess. We have the lungs have lobes, and there is an inferior lobe of both the right and the left lung. Okay. Which one do you want to pick? The primary bronchi? And show me where it goes. Yeah, so it goes to this dot here, right? Isn't that what you're pointing to? Good. The primary bronchi. Bronchi is plural, bronchus is singular, so we have a right and a left main or prime. I use the word main, this uses primary, but a left main bronchus and a right main bronchus. Two primary bronchi.
Those be the fissures?
Show me where the fissures are.
Oh.
Yeah. So the fissures are the divisions between the lobes. So, and they actually have names. This is the horizontal fissure, which is only present on the right lung. And this is the oblique fissure, which is present on both lungs. Horizontal, the names are descriptive, horizontal and oblique. The right lung, for some reason in humans, has 3 lobes, and the left lung has 2.
Would that be the mediastinal surfaces?
That would be the mediastinal surface. So the lungs have lobes, and they have regions, and they have surfaces. And one of the surfaces is the surface that lines the mediastinum, the mediastinal surface. So this whole thing here, in 3 dimensions, is the mediastinal surface. Jasmine, you have the, yeah.
Oh, my house a lot. Okay, so the middle lobe, you were there,
Yeah, so that's the middle lobe.
Nice.
Some of these get easier with time.
Superior lobe?
Superior, upper lobe, so there's upper, middle, and lower, or superior, middle, and inferior. They're all okay with me.
Is that the base?
That is the base. So apex is at the top and the base is at the bottom.
This would be the cardiac impression?
That is the cardiac impression or the cardiac notch. This little cutout of the left lung, not cutout, but it's a little impression for making room for the heart, is the cardiac impression. Okay, so the last one is easy. So surfaces, mediastinal, costal. The costal means ribs. So the surface that is facing the ribs. Remember, this is the lung inside the rib cage. This is the costal surface, and this is the diaphragmatic surface. It's facing the diaphragm. Mediastinal, diaphragmatic, costal. We'll see these in the lab this week. Okay. So this URL is available on the PowerPoint. You can cut and paste it and play this game with your friends. So there is a lot of respiratory anatomy in the nose, and we're going to skip it. So there's a lot of names here, like the names of these lumps in the inside the nostril. This is a conchi. Concus. The grooves are conchi and the lumps are meatises. There's names here that if you become an ear, nose, and throat specialist, you certainly need to know, but we're not going to focus on this. But this game is also available to you to learn from, but you'll notice that these things like sinuses and conchi and tonsils are not on the key concept list. Trachea is. Larynx is. That's about it. Okay, so let's go back to the PowerPoint. I show you that to show you that even though I'm asking you to learn anatomy, I'm not asking you to learn all the anatomy. Okay, so learn the anatomy and the terms that we use in class today and Thursday. So let's talk about airway. The tubes that connect the lungs to the outside world. The airway is highly divided. So we start with one trachea, then we have two main bronchi, or primary bronchi, and those divide into two more bronchi. And they divide and they divide and they divide. All of those tubes are called bronchi until they get very small. So we're now in the getting into smaller and smaller tubes, and those are called bronchioles. Similar terminology to arterioles and venules. Bronchioles are small airway, and those continue to divide until we get into terminal bronchioles and then respiratory bronchioles and alveoli. This is where gas exchange happens. So this is the business end. This is where gas exchange between the air that you've inspired and the blood that is being circulated by the heart through the pulmonary circulation does its exchange. Oxygen is picked up, carbon dioxide is eliminated. All of this plumbing up here is called the conducting zone. It's not, no gas exchange happens here, but it's necessary to bring, it's the plumbing. It brings the air from the nose and mouth to the respiratory zone. So the key words are trachea, primary bronchus, left and right, then a bunch of bronchi, which are not the same as the main or primary bronchus. So if labeling something, you see a dot going to, let's go back to that let's go back to that thing. So you would be able to discriminate between the trachea, the main bronchi, left and right, and then this would just be a bronchus. And this would be a bronchus. And this would be a bronchus. But eventually it's going to go into a respiratory bronchiole and alveoli, which are hard to see because they're microscopic in size. Okay? Clear about the difference between the main bronchus and a bronchus. Okay, so here's a blow-up of a terminal bronchiole, which is part of the conducting airway, and respiratory bronchiole, which is part of the respiratory airway, because gas exchange is happening with these sort of balloon bubbles coming off of the respiratory bronchiole, and then sacs or lumps of alveoli. So this is respiratory, and this is conducting. Obviously, the place where gas exchange happens is surrounded by a network of capillaries. Right? So here is a artery. Remember, it's carrying deoxygenated blood from the heart. So it's an artery, but it's a branch of the pulmonary artery. And then it's forming an arteriole and a network of capillaries, then a venule, and blood is going to return to the left atrium via a branch of the pulmonary vein. These red ribbons in this cartoon are illustrating smooth muscle. Just like the alveoli, just like the arterioles have smooth muscle that control the diameter of arterioles and control where blood flow happens, airway, both the respiratory airway and the conducting airway, has respiratory smooth muscle that control where the air goes. And the mechanisms, we'll talk more about that mechanism later on in the section. But the mechanisms of smooth muscle contraction, you already know a lot about. Calcium, calmodulin, myosin light chain kinase, maybe a GQ coupled receptor. You can apply what you know about contraction of vascular smooth muscle and apply it to airway smooth muscle. Okay, who's got the laser? Okay, this is just a slide, so it's not going to make the happy sound, but pick a name and then point to it on the picture. So there's no dots here. You just show me where it is. Well, there's a couple of asterisks, but show me where the name is. Okay, so is this a bronchus or is that a main bronchus?
Main bronchus.
That is a main bronchus. That's the right main bronchus. Okay? Go ahead and pass it along. You would label this right main bronchus. Yes, that's the trachea. And we can see a little bit of the left main bronchus going off the picture here. Okay? Okay, sure. So here's a bronchus and here's bronchi, and it starts to divide into the meshwork. But that would be, you could point to this and label that a bronchus. Yeah, bronchioles. So maybe at the edge here, you could go to something that isn't the very end, because the very end would be alveoli, and you just go up, that would be a respiratory bronchiole, just up to the edge from the alveoli. Okay, so that's how you would play this game if you're asked to play this game. Any questions about the terminology? So now we're going to turn our attention for a few minutes to histology. In the cardiovascular system, we talked about different cell types. Let's see, let's try to refresh our memory. What were some of the cell types we've talked about in the cardiovascular system? We talked about nodal cells in the SA node that were specialized for pacemaking. We talked about cardiac myocytes in the heart, throughout the heart, that are specialized for contraction, generation of force, pressure. We talked about smooth muscle, that controls the diameter of arterioles. We talked a little bit about Purkinje fibers, but we didn't get into the histology of them. So the concept of different cell types is not new to you. What's going to be new here is that we're going to use micrographs to look at them more carefully than we did in the heart. So the things that you want to know for the quiz are the names of the cell types and what they do and what they look like. Okay, so that's what we're going to work on here. What does each cell type look like, and what does each cell type do? And obviously, mucus is going to be one of those things. So here is a micrograph. This is a photograph of an actual section through an airway. So let's get oriented here. Airway is up here. So this is the air part of the airway, and here is the outside of the airway, connective tissue. Down here would be some other, thing, blood or something. So this is the outside of the airway, and this is the lumen of the airway here. And these are the cells that line the airway. It's a pseudo-stratified epithelium. You don't have to know that. It's an epithelium. It's a layer of cells. That's a word we're going to hear quite a bit from now on, epithelium, because we're going to need that both in the respiratory system, we're going to need that in the kidney, and we're going to need that in the digestive system. An epithelium is a layer of cells that lines a tube of some kind and has important function. So this is an epithelium, and there are two cell types that dominate this epithelium, at least in this picture. There are the ciliated cells, and you can tell that they're ciliated because they have cilia. I mean, you can see. So ciliated cells have cilia on their apical membrane. Apical is the side facing the lumen of the airway. So here are ciliated cells, and one has been cartooned here with, to accentuate the cilia and the nucleus and the shape of the ciliated cell. We'll talk about the function of these cilia. Well, it's right there. It's going to transport mucus. We're going to talk some more about that. In addition to the ciliated cells, there are goblet cells that produce and secrete mucus. And we're going to see goblet cells in the digestive tract as well. And they're called goblet cells in the digestive tract as well. So what you learn in the respiratory system will come in handy when we get to the digestive system. Goblet cells make mucus. So goblet cells make mucus and secrete it into the lumen of the airway. They secrete mucus to moisturize the airway. And the cilia move it. You don't want your mucus to be too stationary. It's unhealthy. It leads to disease. The cilia transport the mucus, moving it up and out of the lungs. And in the process, moving whatever the mucus traps, all the dust that we breathe and viruses and bacteria, the things that come into our body through just breathing air on planet Earth, get trapped in the mucus, and then the cilia move it upstream, and eventually we swallow it. So mucus. We're going to spend a few minutes on mucus. So there's a couple of dramatic videos, which I'll show you, that describe mucus. So I went to the very end of the video. So it turns out that hagfish, a very primitive vertebrate ancestor of human beings, do an amazing thing with mucus. And they produce mucus that's very similar to the mucus that we produce. But they produce it on their skin. That's a hagfish. Believe it or not, that's a vertebrate. Watch what happens when it's disturbed. Produces gallons of this stuff if it's disturbed. And I'll bet you can probably predict why. And the why is shown in the next video.
A shark sees an inviting meal, an eel-like fish with no visible defenses. But the shark is in for a big surprise. Choked with slime oozing from hundreds of pores in the fish's body. Researcher Vincent Zinzan from the Museum of New Zealand Te Papatongarea and his colleagues went near New Zealand's Great Barrier Island and dropped remote cameras. Attached to the apparatus are blue lights to be as unobtrusive as possible and a bait bag. Here, at more than a 2,000-foot depth, some of the first to attack the bait were hagfish, also known as snot eels. At one point, a kite-fin shark, uninterested in the bait, instead prefers the hagfish, but gets a mouthful of slime. The slime fills the shark's mouth, clogs its gills, and chokes it. The hagfish were seen repeatedly bitten by larger fish, all retreating with the same result. Here, a cat shark seems to be fine in biting down on the hagfish, but eventually turns away as it too is gagged by slime.
Okay, so you get the idea. It's protected. Hagfish, 500 million years ago, evolved a sliming mechanism for protection. We have maintained that sliming mechanism for the protection of our airway, and it turns out for also protective of our digestive tract. So mucus is a critical, it's an important part of our respiratory system. Okay, so you can watch these videos. Show your friends the amazing things about mucus. Okay, so let's do a clicker about mucus. Okay, so mucus is a collection of proteins. They're called mucins, and they are glycoproteins. This is using a word that you heard about in self-viz. These are sugar-coated proteins that absorb a lot of water and make a gel, like well, you saw the mucus. It's a gel-like complex, and it's a gel that coats the inside of the airways. I guess number C is up for debate. You can just define whether it's gross. It is not green. It is colorless. The greenness of snot is that's an evidence of infection. Healthy mucus is white or colorless. Okay, what's the function of mucus? I mentioned this in class or a few minutes ago. Now let's see how well you were paying attention. But also just figuring it out. What is the function of mucus? So I'm being a little bit coy here. It certainly traps dust and pathogens. It is also to moisturize, to humidify the air, and to moisturize the airway. So these are the true statements. B, mucus doesn't transport. What cilia do the transport function of mucus. So mucus, the function of mucus is to trap and humidify. The function of cilia is to transport the stuff that the mucus traps. And I mean, this is a true statement, but it's not a function. Okay, so let's do some more histology. This is a scanning electron micrograph of an airway. And this is looking down on the apical or luminal surface of the airway. So we're looking at the epithelium from the lumen of the airway. We're not seeing the connective tissue underneath. And so you can see, let's see, G is a goblet cell. Where is it? There's a G. So these things here are goblet cells. Here is a goblet cell caught in the process of secreting mucus. The mucus was, so if I go back a couple of slides, notice that the goblet cells are chock full of these granules, right? So here is a ciliate cell, but these are all goblet cells. They're full of granules, secretory granules. These are secretory vesicles that are full of the mucins. And they're ready to exocytose those secretory granules into the apical membrane, or into the airway, or onto the surface of the airway. And so here we see one doing exactly that. Massive fusion of those secretory vesicles onto the surface of the airway. These are cilia. And in addition, there is one other type of cells called the brush cells that they're sensory cells. So you know that if you inhale something that is irritating, you cough or you sneeze, it's a reflex to try to get rid of whatever it is you just inhaled. And sensory cells, like the brush cells, are important for that sensory modality, sensing an irritation that triggers a cough or a sneeze. So those are the main cells that we're going to talk about. What does each cell type do? This is what you want to learn. Goblet cells make, store, and secrete mucus. Ciliated cells move the mucus. And brush cells are sensory cells. So one question is, well, how do these fine hairs move mucus, which is it's a hydrated gel? The answer is that the cilia have cytoskeleton inside. So the cilia are, I'm going to go back a few slides again. The cilia are extensions of the apical membrane. So they're not hairs. They're coated with a bilayer. And inside them are cytoskeleton. And so So, and there is cytoskeletal movement. Myosin, well, it's not actin and myosin, it's kinesin, it's microtubule based, not actin and myosin based cytoskeletal movement. You remember from cell phys that we had microtubules and we had kinesins and dyneins moving on microtubules? Well, that same sort of function is happening inside cilia. And so when these cilia move, they kind of beat. So they do like a hula dance. They sweep and then sweep. And they work. They don't synchronize, but they're all doing movement in the same direction. So you can imagine, right? Which way is, so if I'm a ciliated cell and I'm doing something that looks like this, which way is the cilia, which way is the mucus moving? Moving that way, because I'm rowing it that way, and then not rowing it the other way. So if the cilia did this, it wouldn't be very productive for it would move the mucus around, but it wouldn't be net movement. It would just be moving it back and forth, which isn't what we've evolved to do. We've evolved to move the mucus up the airway to the throat, where unconsciously we swallow it. And remember, the mucus does the job of trapping and hydrating, and the cilia do the job of moving it. Okay, another clicker. Okay, I'm seeing a lot of correct answers because I just told you the answer. The answer is A. And what is the function of cilia? The answer is B. Okay, good. You're paying attention. We're learning about mucus and cilia and airway. It's all good. So now we're going to get to the business end of the respiratory system. Well, no, that's not a good way to say it, right? The conducting airway and mucus and cilia is all very important. But now we're going to get to the end of the airway, the dead end, where these sacs of alveoli are and where gas exchange happens. Okay, so I'll show you a few more micrographs. We'll see this structure, then we'll take a break. So we're zooming in on this microscopic structure of the airway. So here is a scanning electron micrograph from a histology book that is looking, so we are standing in a terminal bronchiole and looking down into a sac of alveoli. So we are standing inside here and looking down this way. And you can see that it's divided into these chambers. They're microscopic in size. Each one is only 200 microns in diameter. Remember, a cell is maybe one cell with 20 or so, 10 to 20 microns. So the dimension of a cell is 10 to 20 microns. So these are only, slightly bigger than individual cells. And they're tiny, but there's billions of them. Well, yeah, millions of them. There's millions of them. And so the millions of tiny surface sacs provide a large surface area for gas exchange. It's about the size of a tennis court. We need that surface for gas exchange. Emphysema is a disease where the alveoli are destroyed, and people die of emphysema because they don't have enough surface area to do the necessary gas exchange. Yes?
So is the alveoli the ball, like
Yeah, the alveoli are the ball, so one alveolus would be one of these little bubbles or balls, and alveoli is the plural, and you would refer to this clump of alveoli, or an alveoli sac. I don't teach Latin, so I'm not gonna say, if you point to this and say that's an alveolus, and you point to this and say these are alveoli, that's the same answer.
Yeah, but like if that's real picture, what's the alveoli?
Oh. This lump right here would be one, I can't see what's behind it, but this would be an alveolus. This is the bubble, or the ball-shaped thing coming off of the tube. Did that answer your question?
Yeah, but like there's another arrow right now. But
So this is pointing, I don't know why it's pointing to these dots. The whole thing provides, so this, maybe there was something else in the histology book that it was pointing to these for some other reason. This is referring to the whole thing, the surface area, the large surface area. This, I don't know what this is. We'll write it out. So here's another scanning electron micrograph of alveoli. Here's one alveolus. Here's another alveolus. Here's one up there. So these are the air-filled sacs, the alveoli, and you can see that they are surrounded by the network of capillaries. So all of these circles are capillaries that have been cut in cross-section. And you can see this intimate relationship between the air in alveoli, the gases in alveoli, and the blood in the pulmonary circulation. And that's, again, essential. That huge surface area is essential for gas exchange. So let's take a break, and we'll come back to a cartoon from your text that talks more about this in a sort of cartoon version. Let's continue. So here is a cartoon from your textbook of an alveolar sac at the end of a bronchiole. And there's some things here that are illustrated that I want to highlight. One is that the bronchioles, so notice here, what do you suppose these little pink lines are supposed to represent? Cilia. So, and we don't see any of them in the alveoli. Well, forget about that one right there. We don't see them in the alveoli. So the cilia are present in the conducting airway. They're not present, so even though this is a respiratory bronchiole, it's a respiratory bronchiole because it's got an alveoli or sac attached to it. The way to think about this is that mucus, produced by the goblet cells and moved by the ciliated cells, is part of the conducting airway and not part of the respiratory airway. And that's cartooned by showing these cilia as part of the bronchiole, not part of the alveoli. The alveoli have been specialized for making this extremely thin barrier between air or gas and the blood in the capillaries. Yes? The brush cells are also only in conducting airway. Yeah. That's right. The sensory cells are in conducting airway. So, okay, question. Does the trachea have ciliated cells?
Yes.
Yeah, it must, because it's moving the mucus up the airway to the mouth, so the trachea must have cilia. and it must have ciliated cells. And it also has goblet cells. So all of the conducting airway, from the trachea to the main bronchus to bronchioles, all of the conducting airway has the brush cells for sensation, the goblet cells for the production of mucus, and the ciliated cells for the transport of mucus. But the gas exchange cells, the alveoli, don't. We do not want mucus in the where there's gas exchange. So there are two types of cells that make up the respiratory airway, where gas exchange happens. There's type 1 cells. Where's type 1 illustrated? Well, this is supposed to illustrate a type 1 cell. Okay, here is a nucleus of a type 1 cell that's lining this airway. So this is a type 1 cell, this is a type 1 cell. They are extremely thin. So here is a cell. This is a red blood cell. And this is a cell. This is a type 1 cell, and it's extremely thin. It's smaller than the red blood cell. There is an interstitial space between the airway and the capillary, and there's a basement membrane around the capillary. So there are these thin structures, but they're all extremely thin, and so gas is very close to blood. And we have that big surface area, so we have ample opportunity for gas exchange. Yes? Oh, everything in the lung is all mixed up, so I couldn't tell you what orientation this section is.
Which way are we looking? Are we looking down from top? That's why.
We're sort of looking, we're cutting an airway. So remember, this is a tube. Right here is a tube leading into this sac.
Right.
And we're cutting the whole thing in half. Okay? So this is the tube, this is the sac, and now we're zooming in on this part of the sac where we have air on one side and air on the other side, and this very thin cellular layer. So here's the air on the bottom, here's the air on the top, but the point is the layer of cells that make the alveolus, mostly the type 1 cells, and then the cells that line the capillary. Remember, we've got endothelial cells and a basement membrane. Here's the endothelial cell, here's the basement membrane, here's an interstitium. All of those spaces are smaller than a cell. Smaller than a red blood cell, anyway. They're all cellular. I mean, this is a cell that is thinner than a red blood cell. This is a cell that is thinner than a red blood cell. So the thinness of this separation between blood and air is critical. There's another type of cell in the alveolus called the type 2 cell. So the names are easy, type 1 and type 2. But their function is very different. Type 1 cells maintain this very thin barrier, and type 2 cells secrete something called surfactant. It's not mucus. It's a secretory process, but in this case, it's secreting surfactant. We're going to start with surfactant on Thursday. I'm not going to talk too much about surfactant, but you're going to see it in the lab on Thursday, and we'll talk about its function in class on Thursday. Right now, I'm just introducing the idea that the type 2 cells in the gas exchange areas, the alveoli, are the cells that make and secrete surfactant. So here is a transmission electron micrograph. So this is now huge magnification. This is blood. This is air. This is a red blood cell. right? This whole, I mean, we're only seeing a piece of it, so that tells you how zoomed in we are. This is half a micron. So P1 stands for type 1 cells. This is a type 1 cell. Some books call it pneumocytes. So it's a type 1 pneumocyte. It's a type 1 cell. You can see, extremely thin. Here's the basement membrane. Here is the endothelium. Here is the endothelium of the capillary. So all of this is about half a micron in size. And remember I said cells are 10 to 20 microns, so this is extremely thin barrier. Oh, there it is, P1, type 1 alveolar cell. And so I'm going to end with this. I think this is the last slide. So the point is, why have I been dwelling on this thinness of this barrier? The thinness of the barrier is essential because the force that moves gases is just diffusion. You may remember from self-phys that oxygen and carbon dioxide are some of the molecules that simply diffuse through membranes. Ethanol is another one. So oxygen, we don't have oxygen pumps. We don't have carbon dioxide pumps. We rely on simple diffusion to move oxygen and carbon dioxide from air to blood or vice versa. So now I'm not going to test you on the biophysics, but there's an equation, biologists and biophysicists can define an equation. So this is Fick's law. Just if those of you who do biophysics and you study diffusion of anything, you'll study Fick's law. So it's let's just interpret this for a minute. It's a constant, some diffusion constant, times the surface area of the thing of the things that separate the two spaces where the gases are exchanged, times the gradient of the molecule that's doing the diffusing. So, and J is just the rate of diffusion. So J is a flow term, D is a constant, A is the surface area, and delta C over delta X is the concentration gradient. And so you can see, here's the biology of it. The diffusion is, well, let's go to some of the things let's go to this one first. The diffusion, the rate of carbon dioxide or oxygen diffusion in an airway, is proportional to the surface area. And this is why we have evolved a very large surface area that we pack inside our lungs. The rate of diffusion is proportional to the concentration differential, if that makes sense. So oxygen in the air is going to be, the concentration of oxygen in air is going to be higher than the concentration of oxygen in the blood, at least at the beginning of this process. By the end of the process, they might have gotten close to equilibrating because it's just passive diffusion. Delta X is the thickness of the barrier. So the diffusion rate is inversely proportional to the thickness. So the thicker the barrier, the worse the diffusion. The larger this term, the smaller the diffusion term. So the thinness, the thickness of the barrier. A thin barrier makes for a rapid diffusion. So there are going to be a couple of equations in respiratory physiology, just like there was a major equation in cardiovascular physiology. Here's one. And the diffusion constant is proportional to things like the size of the molecule. Well, carbon dioxide and oxygen are both very small, so they tend to diffuse fast. It's proportional to, It's proportional to temperature, inversely proportional to size, and inversely proportional to the viscosity of the fluid, right? You can imagine that something diffusing through honey is slower than something diffusing through water. And in the spaces, and so you can imagine that something diffusing through mucus is slow compared to something diffusing through water. And the fact is that there's the reason why we don't have mucus in our respiratory airway, is because it would impair the diffusion of gases. We have a very thin layer of water and surfactant. And we'll see more about surfactant on Thursday. So diffusion is important here. And we're talking about diffusion of gases between air and blood, or between blood and air. And we'll talk specifically about the transport of oxygen and CO2 before the end of the section of the course. Any questions about diffusion or any of the anatomy that we've talked about so far today? Yeah.
So delta X is a sticky. Why is that delta X?
Oh, we could call it X. It's just, I was trying to draw this as a gradient. So we could just call this X if you want. If that makes it easier for you, call it X. The C is a concentration gradient, and the X is the thickness of the thing that separates high concentration from the low concentration. That's fine. Biophysicists will turn this into a differential equation and solve it for